So hello and a very warm welcome to Nature Water Talks. I'm Fabio Pulizzi, I'm the Chief Editor of Nature Water, and I'm delighted to be the host for today's event. So Nature Water Talks is a series of online conversations that was launched in the summer of 2022 by the editorial team of Nature Water. And the idea behind this series is very simple. The idea is to explore some pressing water-related issues by speaking directly with the experts, so going beyond just what we can learn by reading their papers. So today we're looking into a topic that sits right at the heart of environmental science and the sustainability of water resources, which is water pollution and the advanced treatment for water pollution. So, and we are especially pleased to be collaborating with our colleagues at Communications, Earth and Environment. And particularly, I'd like to mention Somaparna Ghosh, who is here with us and who is curating an open collection on this subject, and she will tell us just a few words about it. So water pollution is, of course, and unfortunately, a very vast and complex challenge. So for today, we only focusing, only is a small word, but it actually means quite a lot because, as I said, it's a very vast field. But we're focusing on two types of contaminants. And they're the two types of contaminants that have somehow captured the attention, not just of scientists, but of society more generally. And these are microplastic and nanoplastic by extension, and PFAS compounds, the ones that are often referred to as forever chemicals. So we're very grateful to have three leading experts in these fields today with us. One is Denise Mitrano, who was recently transitioned from ETH Zurich to Nestlé Switzerland. The other is Denis O'Carroll, who is from University of New South Wales, and Yang from the Clarkson University in the US. So each of them will begin with a short talk introducing their work and their field of research. And then we move to a panel discussion that will be moderated by Somaparna and my colleague from Nature Water, Shujuan Zhang. But before that, Somaparna will just give us a couple of minutes introduction to what the collection is and how it connects to today's talk. And just a quick reminder that the Nature Water Talks are actually hosted by the SDG Talks, and you can access and register to them by following this QR code, and so that you can be updated on future events. The webinar is recorded and will appear on our website and on the SDG Talks website in a few hours. And if you want to ask a question at any time, use the chat box, and then we'll address it to our speakers when it's appropriate. But now I'm going to stop talking. I'll give the word to Somaparna, and Somaparna will just talk to us for a few minutes about the collection.
Thanks, Fabio, for the introduction. Welcome, everyone. I am Somaparna, and I am the Associate Editor with Communications, Art and Environment, also a consulting editor with Communication Sustainability. So I will walk you through a brief introduction of our newly launched collection on water pollution and advanced treatment processes. So talking about the scope of the collection, as Fabio said, and we already know and agree that the treatment of water and wastewater is a critical area of research given the increasing global challenges associated with the water scarcity, pollution, and public health. So we have launched this cross-journal collection to highlight the important and trending subject areas such as emerging contaminants and the advanced processes to remove these contaminants from water, and to bring together articles across other nature portfolio journals in one place, and also to help with communication across the specialist field. So talking about what we are looking for high quality research on the areas, but not limited to emerging water contaminants and their impacts on ecology, environment, and human health. Development of novel advanced water and wastewater treatment processes and their applications at both local, regional, and global scale. Studies at the intersection of the advanced treatment technologies in the context of resource recovery, and circular economic principles, and sustainable management of water sanitation for all in line with the SDG goal 6. Next slide, please, Fabio. So now few details about the collection. The submission is open. You can submit your manuscript right away to us. The deadline as of now is 28 November, which can be extended for a couple of months. So don't worry if you don't have your paper ready right now, you still have time to submit with us. The participating journals are Communications, Art and Environment, Communication Sustainability, Nature Water, Nature Communications, Scientific and Scientific Reports. So this is the QR code that you can scan and it will direct you to the collection website. And if you want to know more about the goal and scope of our journals and about this collection, please feel free to reach out to us. And if you are interested, we can organize a research talk or webinar on the journals and about the collection. That's all about me. Thanks very much. I will pass it to Denis. The floor is yours, Denis. Thank you.
Hi, good afternoon, good morning from wherever everybody is around the world today. Thanks for joining this seminar series. My name is Denise Mitrano. I have just recently moved to Nestlé Research here in Switzerland, but prior to this, I was Assistant Professor of Environmental Chemistry of Anthropogenic Materials at ETH Zurich, and that is the work that I'll be presenting today. So just to point out that nothing that I'm presenting today is affiliated with Nestlé Research. So kicking off this series about understanding pollution in aquatic systems, the topic that we were studying is nano and microplastics in aquatic systems. So while many of you may have heard this term before, if you're not researching the topic, you might not exactly know what nano and microplastics are. So this many different sources and types of materials that fit together to form this puzzle of what are nano and microplastic pollution in the environment. Most of these materials come from the slow degradation of plastic waste that is unintentionally put into the environment to form particles that are lower than 5 millimeters in size for microplastics or less than 1 micron for nanoplastics. Aside from this degradation of mismanaged plastic waste, we also have the mechanical stress on types of items that we use every day, such as tireware or textiles, or degradation of plastics which are intentionally put in the atmosphere, such as mulching films when used for plastic culture. A small proportion of nano and microplastics are primary microplastics. Those are put directly in different types of consumer products and for different types of applications that the materials are directly formed at that size range and then may also leak into the environment through various routes. So where the research is going overall, we need to look at how to frame this issue of microplastics and nanoplastics in the environment. When we take a microplastic-centric view, a lot of researchers, including ourselves for some of our work, are trying to enumerate the amount of nano and microplastics that are in the environment. So how many are there? What are their properties, their physiochemical structure? How are they transported from A to B? And if these materials are ingested by organisms, aquatic organisms, soil organisms, or human, are there any ill effects? In contrast to this microplastic-centric view, our lab was also looking at an environmental systems view of nano and microplastics. So really, how are these materials interacting with natural systems? Are there any impacts on biogeochemical cycles? And are there any indirect effects on health? This takes a more holistic One Health perspective, which can also give us a better idea about really what are the implications of these materials when they get into environmental systems. So for the rest of this talk today, we'll look at nano and microplastics in water, even though my lab group had more different types of research than just the aquatic environment. The first aspect I'd like to share with you is more about how they get into the environment and then once they're settling in aquatic water bodies and their implications there. Since this webinar is also about the advanced treatment, there's also information on removal and wastewater in drinking water treatment plants. So when we think about microplastics getting into freshwater systems, there are a lot of different entry pathways into the aquatic environment. And then once they're there, they may move and behave in different ways, either alone or in conjunction with natural particles, be that organic, mineral, or biological particles like algae. So depending on the density and size of the plastic, it may float or sink and settle. It can aggregate with other natural particles, which may enhance the settling dynamics, or this can also impact the lateral transport of these materials when there is flow with the water. For the study that I'd like to present today, we're mostly looking at the heteroaggregation and how the interaction between microplastics and these natural particles influence their settling rate over time. For this, we need to take a little bit closer look about which types of natural particles are important in these freshwater systems. Here, I've highlighted freshwater snow, which is a natural mixture of algae, particulate organic matter, and other trace inorganic materials that will flocculate in these natural water bodies. So even if something from a distance looks clear, I'm sure when you've all been swimming, you've seen a little bit of fuzziness in some of the waters, and this is this freshwater snow. So this is very key in biogeochemical cycling of nutrients from the water surface to the sediments, because these algae grow at the surface and then bring their nutrients with them down through the water profile and over depth. So to be able to understand how microplastics interact with these freshwater snow, we assess the settling rate of microplastics of different sizes and densities, either alone or in conjunction and heteroaggregation with freshwater snow. We then aggregate this freshwater snow with the microplastics and again assess how the settling rate changes of these heteroaggregates. With this type of workflow, we're able to see a few different things. Firstly, our standard models to be able to assess the settling rate of particles, for example, of natural particles, relevant for microplastics, and does this actually change when they're aggregated with freshwater snow? Having this information would help us to be able to constrain a bit more the different fate models of microplastics in freshwater systems. So how we actually did this in a laboratory setting, we had a large column of 1.5 meters high, which was illuminated by a laser light. We would put in fluorescent microplastics or aggregates of microplastics with our freshwater snow and let them settle over the column through time. To be able to capture their settling rate, we had a series of cameras with different types of optical filters to be able to look in a 3D space how the materials were moving. This allowed us to track thousands of particles settling simultaneously under different conditions and different particle types with different sizes, shapes, and polymer chemistry. So the influence of microplastics really not only changes in their settling dynamics based on the materials themselves, but if they're aggregated with this freshwater snow. So when we look first at high density microplastics, such as those from PET, we looked at fragments and fibers. We can see on the y-axis the settling velocity and on the x-axis the different size of microplastics. As you might anticipate, the larger the size of the fragments, because they have a larger mass, also settle faster than the smaller ones. And those particles which are more dense also settle more quickly. In contrast, the morphology of the material is really important, because when we look at fibers, which have the same overall mass as a large microplastic size, they settle significantly slower because of how they orient themselves in the water column. Now, when we look at this aggregation with freshwater snow, now in the box on the right, we see freshwater snow in brown. This is the natural settling rate of the freshwater snow, but we see that the heteroaggregates are settling faster than the freshwater snow alone, indicating that the microplastics can act as a ballast to bring this freshwater snow more quickly from the water surface to below. When we also compare this to the typical settling rates, which would be predicted from example Stokes' law when looking at the natural behavior of particles, we see that these settling rates that are presented here are actually much slower than would be anticipated if we had a particle of the same mass and the same size of natural composition. So this information can also help us constrain fate models to be able to understand how quickly and how far particles may be able to go in an aquatic environment. So collectively, this project has shown that microplastics and freshwater snow interactions influence the respective settling rates. Especially when microplastics can act as a ballast for the freshwater snow. The microplastic morphology also needs to be considered when assessing the settling rate because of the different shape of the fragments, and the fibers will influence how fast they will settle compared to more spherical particles that are of natural origin. Ultimately, the impact of microplastics in the environment could also impact nutrient cycling by increasing the settling rate of freshwater snow. Although here it has to really be noted that this has to be at quite a high concentration of microplastics, because even though we find microplastics everywhere around the globe, still they are a relatively low concentration when we're comparing these to other natural particles, which are already in aquatic systems. Moving on from these natural systems to a more engineered environment, we can also look at the removal during wastewater treatment. So microplastics and nanoplastics are often directly released to wastewater, especially when they're used in consumer products. And we wanted to be able to assess if conventional wastewater treatment plants could actually remove them, and at which stages of wastewater treatment is the most efficient for particle removal, and if this is particle size dependent. So here we tested both nanoplastics and microplastic fibers to see how they are removed during water treatment. If you're not very familiar with a wastewater treatment plant, here's a quick schematic where we have raw wastewater that is coming into the plant, have a primary clarifier, which already removes about a large proportion of the settleable solids, then moving on to the biological treatment steps, and a secondary clarifier, which again moves more of the settleable solids before the water is released into receiving water bodies. To make this problem a little bit easier for us to start, we didn't start directly in the wastewater treatment plant, but rather used batch tests to look at the heteroaggregation, again, between microplastics and nanoplastics, and the sludge flocks that are in this mixed liquor in the biological treatment steps. So again, you can think about it in the same type of context that we use in the heteroaggregation study, which I presented earlier. We then would mix for different amounts of time, allow this to settle, and then sequentially sample through the supernatant into the sludge to be able to see where the nano and microplastics were over time. As we can see, only presenting here the nanoplastics, but the same trend was also shown for the microplastic fibers, that there were very few particles that were left in the supernatant, what would eventually be going to receiving waters. So this was the same for both the plastics and the total suspended solids, the organic matter. And as we increase the amount of total suspended solids, as we go lower in the sludge layer, we also have more plastics that are present there. This shows a strong indication that they have heteroaggregation between the different plastic particles and the sludge solids. When we move again back to the longer-term assessment of nano and microplastics in a wastewater treatment plant, we worked at a pilot scale wastewater treatment plant that looked just at this portion of the treatment process, and again wanted to zoom in on these biological treatment steps and the secondary clarifier to see how many particles would be removed from the wastewater treatment. So we could put our particles in at the beginning of this treatment step and then check how much was in the effluents and how much was coming through the settleable solids. So when we look at this graph, on the y-axis, we have the concentration of nanoplastics, and over time, you can see that we ran this plant for about 2 months. We had initially high dosing of both nanoplastics and microplastic fibers, and then had a lower dose of a continuous feed over time before stopping the input of nano and microplastics into the system. Over this entire time, about 95% of the nanoplastics and microplastics fibers were found in the sludge, which is in the orange triangles. And we can see that the plastic really follows the sludge model, which is in the black dashed lines, indicating that the particles in the system either aggregates to the sludge and is removed with the sludge, or behaves essentially like the sludge particles anyways. So again, we had quite a high removal of nanoplastics in these systems. Going quite quickly to the removal during drinking water treatment plants, microplastics and nanoplastics are in many drinking water sources, as we see in freshwater environments. But we wanted to assess if conventional drinking water treatment plants could actually remove them, and again, at which stages of drinking water treatment are most responsible, or if there are different operational conditions that are most efficient for their removal. So here again, we look in different types of laboratory scale columns and working at a pilot scale water supply company to be able to assess the removal, especially in this case of nanoplastics, at different scales and at different timelines. Using the laboratory and pilot scale systems together, we could then model what would eventually happen at a larger scale, typical drinking water treatment chain. So when we look at the simulated performance of the drinking water treatment chain, we don't have any removal of nanoplastics in an ozonation step because it's not a particle removal system. But looking at the rapid sand filtration, activated carbon filtration, and slow sand filtration, nanoplastics were cumulatively very effectively removed from the entire drinking water treatment chain, and especially the slow sand filter was very important. So this is important to note because not all drinking water treatment plants are set up the same way, and they can have different operational steps. So identifying those different treatment steps, which are most important for particle removal, would also help identify how to keep our drinking water safe in the future. So with that, I will close my presentation, looking forward to the presentations of the other speakers and to the discussion session at the end. Thank you.
Thank you very much, Denise. And Dennis, I'll let you go next. Let me mute you.
Wonderful. Can you see my screen okay?
Yes, all fine.
Great.
Thank you.
Great. Thank you. And great talk, Denise. So my name is Denis O'Carroll. I'm at the University of New South Wales in Sydney, Australia. I'm going to talk today a bit about PFAS, some of the challenges we have with working with PFAS and PFAS research, the sources of PFAS to the environment, and really very briefly touch on treatment technology that we're working on. So PFAS are pervasive in our daily lives, much like plastic, of course, and in a range of consumer products. And there's over 14,000 PFAS or distinct PFAS molecules. I'm going to talk a little bit about the different PFAS classes that we have. And in the colors, it's a little bit hard to see here, perhaps, but the colors just denote the different PFAS classes in firefighting foam, AFFFs, as well as consumer products on the right-hand side. So it could be coatings, cosmetics, let's say textiles. And you can see right here that there's a range of colors, and they would denote PFAS classes. So the first one could be perfluorocarboxylic acids, and PFOA is one of those, very, very common and well-regulated around the world. Perfluorosulfonic acids in orange, and you can see there's a range of orange in firefighting foams and some a little bit in consumer products. Fluorotellular sulfonates, you can see quite a bit of green here in the firefighting foams and a little bit in coatings and in some paints. Fossa, you can see it quite a lot again in firefighting foams. And then the last one, this is sort of a pet one that we work a little bit in, is fluorotellular alcohols. And you can see really a lot of fluorotellular alcohols in consumer products and in coatings. And I would, I'm not really going to get into it too much, but fluorotellular alcohols would be a PFAS that isn't really well studied in many environmental engineering laboratories, research laboratories, because it typically requires a gas chromatograph as opposed to the other classes that we would use a liquid chromatograph. So I'm going to quickly go through a brief timeline of PFAS regulations, and that this represents some of the challenge of working with PFAS, is that really only in 2009 did we PFAS started to be regulated. Certainly, maybe about a decade earlier, or sort of society started to be concerned about PFAS, but it was really until 2009 there wasn't much regulation. And in 2009, PFAS was subject to the Stockholm Convention, and PFOA, the U.S. EPA, started to regulate it or provide a guidance in drinking water. We fast forward to, let's say, 2016 and 2019. You can see that PFOA was added to the Stockholm Convention. Canada, where I'm originally from, started to provide guidance for a lot more PFAS at a range of concentrations here. And then in 2022 and 2023 and 2024, Canada, the European Union started to really regulate all PFAS. So in the last 15 years, then we could see from really being concerned about PFAS and PFOA 15 years ago, and now potentially concerned with all PFAS potentially, and that represents over 14,000 PFAS. So quite a lot. The US EPA, it depends on this may be repealed, but they've now provided guidance on more PFAS. China has guidance on PFAS, and more PFAS are being considered in the Stockholm Convention. So it really represents a challenge, certainly for water utilities, and a little bit this came out of the American Chemical Society last year. But because of those new guidelines, up to around 5,000 utilities in the United States are going to have to upgrade their water treatment system. That's going to represent a cost of potentially $15 billion. And 3M is who invented PFAS last decade or early last century, has to pay over $10 billion. And Calgon, who supplies activated carbon, has a vast market now, because activated carbon is one way to remove PFAS. So we last year published a study in Nature Geoscience, and is mostly led by Diana, an outstanding PhD student of mine, and with strong support of Daniel here. And we looked at PFAS in our global waters, and we didn't measure it, but we collected literature data for on the order of 50,000 data points from around the world. And that's just water samples for PFAS concentrations in groundwater and in surface water. And what we did is we benchmarked it against international drinking water guidance. And this here is a map of the different points. And 100 nanograms per liter is the guidance in the European Union at the moment. So and that's a sum of 20 PFAS. And you can see any circle here that is circled with a line indicates that it's over the limit. And the red circles indicate that it's from the source is a firefighting source. And if it's a black source, it would denote that it's a non-firefighting source. So then we compared the different sources of water that we collected. And let's say a surface water from a firefighting source, and here there's on the order of 3,000 samples, and compared it to international drinking water guidelines. Let's say the US EPA guidance, or Health Canada, or the European Union. And what you can see is surface water from a firefighting site with a known source. Upwards typically over 50% of these surface water samples exceed these drinking water guidance, which is unsurprising. Similar for groundwater. If you have a surface water from a non-firefighting source, but that could be a landfill or a wastewater treatment plant, again, a significant number of those, or important number of those samples, would exceed guidance values, as would groundwater. But when we looked at, let's say you went into an urban area and took a sample from an urban stream or groundwater, that, when you don't know if there's a source of contamination, to our surprise, a reasonable number of these samples, let's say on the order of 15% or up to 40% for the US EPA PFOA limit, exceed the surface waters exceed these drinking water guides. You wouldn't, for many reasons, but from a PFAS perspective, you wouldn't want to drink these waters. And for groundwater sources, the exceedance is even higher. So what this would suggest is that PFAS is pervasive in our environment, and if we don't know if water is contaminated over regulations. Even if there's an unknown source, so we need to check. So a big question for us is, where does the PFAS come from and where does it go in the environment? For us as in society, when we flush our toilet, water, as Denise says, goes to a wastewater treatment plant, and we often take the biosolids from wastewater treatment plants and we apply them to land. So if we have PFAS in our wastewater, and we do, it can be applied to land. Or when we throw away waste products, let's say like used carpets, they end up in a landfill, and they can be sources of PFAS to the environment. So a big question that we're trying to grapple with now is how do we do source control? How do we remove the flux of PFAS to our wastewater treatment plants such that we don't apply it to land? We don't inadvertently contaminate our lands. And a real challenge at the moment is figuring out what are the mass fluxes of these different potential sources. PFAS could be in food containers, it could be in carpets. Of course, it's historically been well known in firefighting foam, and we used to use it quite a bit in as water repellency in clothing. So another issue that we're grappling with is, and Denise did a great job saying that we in our wastewater treatment plant, we have an effluent and a biosolid stream, and we really want to use these two streams, and certainly in Australia, which is in many regards water poor. We want to reuse the effluent, and we want a beneficial reuse, and our land is often quite challenged, and we need the nutrients. So we want to reuse these two sources of potential nutrients in water and have a circular economy. So how do we do that? And one thought is, can we have a third stream, a third effluent type stream with concentrated PFAS, and that we can figure out what to do with. So many of the this is looking at international soil guidance and looking at the extent to which biosolids exceed these soil government criteria, and you can see that a large number of our biosolids would exceed soil criteria, which is a problem because then we don't want to apply biosolids to land, and if we don't apply the biosolids to land, it becomes a challenge. What do we do with the biosolids? Do we incinerate them? Do we put them in a landfill? And these are all costly processes and also have a significant carbon footprint. So in our lab, we're working to develop PFAS treatment technologies, and I'm part of a large team at the University of New South Wales working on this. And essentially, one of the treatment trains that we're working on is having a reactor with activated carbon. And on that activated carbon, we graft on redox active catalysts, and we're working to develop the right type of catalyst such that we can destroy the PFAS and defluorinate the PFAS. And with that, it's a redox type process. So we have to figure out where do we get our electrons from, which it does represent a challenge. It's sort of like putting together a jigsaw puzzle because we have all kinds of different PFAS sources and types, as I talked about. We're trying to work on figuring out the right type of catalyst, so the right type of properties, working on the different electron sources, and ideally it would be microbiology, and as well as figuring out the right reactor design, because that does have operational challenges. With that, maybe I won't get into the summary, but I'll just thank everybody and look forward to the other speakers in the chat.
Thanks so much, Dennis. Very, very interesting talk. Yang, so I'll ask you to unmute yourself.
All righty. Okay. The slide looks okay to you?
Perfect.
Okay. Thank you so much for the opportunity. My name is Yang. I came from Clarkson University in upstate New York of the United States. So today I'm gonna, continue the discussion about the treatment technology. We will address complete PFAS destruction in water and also in the solid phase. And before we go any further, I would like to acknowledge my talented student and my fantastic collaborator, especially Professor Jinyong Liu from UC Riverside, and my other collaborator from Yale and New Jersey Institute of Technology. And we can't make this far without all this general support from NSF and DOD and DOE. So we keep talking about treating PFAS is very challenging. I think there are three challenges ahead. One of course, it's it's a family of chemical with different structure, and the different structure leads to very different physical chemical property. And the second challenge is that it usually has a very low concentration. If you imagine you want to remove like PPT or PPP level of PFAS in water, that means you are basically picking out one PFAS molecule versus a majority of water molecule. And as I will mention later, water molecule tends to receive or lose electron first prior to PFAS destruction. And the third challenge is there are different type of definition about PFAS treatment. You can remove it by absorption, or you can partially destroy the structure so that the parent compound won't be detected, or I would like to follow a even higher bar of let's say 100% defluorination. That means all the organic fluorine should be knocked off and recover as a inorganic fluoride, so that we make sure that there's no further secondary pollution occurred. So let's say if we develop a method to destroy PFAS, and what would be the energy requirement? As a chemist, you might already know there's an equation called delta G equals NEF. That means all the Gibbs free energy can be correlated or can be described by reduction potential. So if we look at this fantastic diagram, in order to oxidize PFOA or PFOS, you need about 2.5 to 3 volts. In order to reduce PFAS, you need about -2 or -3 volts. So basically, roughly, you need about 2 to 3 volts electrical potential. But keep in mind that water will only be stable around 1.23 volts. So in this case, whatever redox technology you apply in water to treat PFAS, water will be the first one to react. And it will be the major competitor for electron. Not to mention other matrix like iron, a redox active ion, or natural organic matter. So it is very challenging to create a required high enough thermal criteria for PFAS destruction, and also selectively deliver this energy to PFAS molecule. So if we further dive into the electron efficiency, let's just look at this problem as a simple, chemistry problem. So if we want to completely mineralize PFAS, recovery as fluoride, we know the stoichiometric ratio of the electron is going to generate it. And if we take our experimental setup as an example, if we apply this much electron to destroy PFOS, then we simply compare the required molar amount of electron to mineralize PFAS versus the total electron applied to the system through our electrochemical oxidation cell. We can have this conclusion. This is just a very rough calculation. If we start with 1 ppm of like PFOS, we might, if we are lucky enough, we might have almost close, more than 90% electron efficiency. That's not bad. That indicates a pretty high selectivity. But if we aim to treat nanogram or microgram per liter of PFAS, then our electron efficiency will drop significantly down to a very, very low percentage. That means the treatment process won't be efficient at all. So this is just my thought. I'm happy to hear a different opinion. So I believe that if we are going to treat PFAS in low concentration around PPT or PPB level, then the water treatment process won't be very cost effective. The water treatment process could play a more reasonable role in treating concentrate. So in order to address that, a reasonable strategy would be using separation technology to pre-concentrate all these PFAS so that they can feed to a non-potable water treatment process to destroy the PFAS. And of course, in some cases, we need a solid waste management approach to get rid of those spent resin and spent GAC. Either way, our goal should be achieving 100% defluorination. Again, that means converting all the organic fluorine from PFAS to fluoride. All right, then I'm gonna talk about the toolbox we have. Keep in mind that there are many fantastic technology beyond the scope of this talk, so I'm just gonna focus on what I've been doing. So in our lab, we have been doing electrochemical oxidation. This is a technology that use anode to force an electron to transfer from PFAS to the electrode, so that the PFAS structure can be destabilized and subject to self-destruction. Another technology we have been investigating is a photochemical reduction process. In this case, you have sulfide added to a UV reactor so that you can generate hydrated electron. In this case, you initiate a photochemical reduction approach, giving an electron to the PFAS. This approach also destabilizes the PFAS structure and leads to either further defluorination or the formation of polyfluorinated structure. So we conduct a comprehensive investigation, and we found that if we apply electrochemical oxidation alone, there are certain PFAS structure cannot achieve 100% defluorination. However, if you put a UV sulfide pretreatment step first, and you kind of do some surgery on the alpha or beta carbon, you can change those perfluorinated structure to polyfluorinated structure, and eventually they can be transformed into a structure that can be completely electrochemically oxidized to fluoride. So we developed a universal technology, and we apply this technology for the treatment of very complicated PFAS mixtures such as AFFF, and we are able to achieve 100% defluorination of AFFF in different water matrix. And this paper was fortunate enough to be published on Nature Water last year. And another topic I would like to talk about is how can we tackle PFAS in solid phase. Obviously, we cannot simply electrify soil like this. So we are inspired by an interesting physical phenomena called piezoelectric phenomena. In this case, if you let's talk about the principle of lighter. So in the case of a lighter, you have a hammer hitting a piezoelectric material, and after receiving this impact force, the piezoelectric material can create a potential, a very high potential that is high enough to ionize air to create spark. So I keep talking about potential because this is the key to initiate all these redox reactions. So if you have a piezoelectric material like this, and you make it a shape deformation about in a 0.08 micrometer, according to this chart, you will be able to create 3,000 volts potential. Remember, we just need 3 volts to destroy PFAS. So the principle of piezoelectric material can be applied for PFAS remediation in my opinion. So we developed this system. So this is called mechanochemical ball mill. And in this setup, you just simply put PFAS contaminated solid along with all this piezoelectric material in the pot, and then start shaking. The stainless steel ball collision will create enough mechanical force, and according to our calculation, the voltage can be as high as 1,000 kilovolts. And then we apply this principle of piezoelectric material assisted ball milling for the destruction of solid PFAS. We achieve 100% defluorination of PFOA and PFOS. And we achieve, we also recently applied this technology to treat the PFAS in the spent sorbents, and 100% defluorination and complete PFAS destruction was also achieved at ambient condition. So this is an emerging area, and I welcome my colleague to have more intensive discussion with me. So these are a series of studies we've been published in the last 2 years. So the last question I'm going to walk you through really quick. Can they be scaled up? The answer is yes. This slide shows you a scale-up system that have been serving in the industry for more than decades. So the commercial product is definitely available. And can the ball milling process be scaled up? In the lab, we use a planetary ball mill, but in the field, actually, there are larger scale ball mill for mineral industry. And in our lab, we are working on the pilot scale demonstration of this ball milling process. And with that, I welcome any question. Thank you.
Thank you, Denise. Thank you, Yang. Thank you, Dennis. So we've heard about microplastic and nanoplastic, fate, and a bit of treatment. We've heard about PFAS, its fate, its dynamics in the environment. We've heard about treatment. So now Somaparna and Shujuan have prepared some points of discussion, particularly to see whether there are commonalities between these two contaminants, but not only these two emerging contaminants, but not only. And then I will remind you again that you can ask questions in the chat. But I will now again stop talking and leave the word to Somaparna and Shujuan.
Thank you, Fabio. Thank you for our three presenters. The talk is wonderful. So, generally we are talking about the two of the most important, I think, emerging contaminant in our environment right now. And when we talk about them, we talk about micro and nanoplastic and PFAS. For example, we see that plastics, they come in different shapes and sizes. So I know Denise talked some on those there regarding their fate with the different shapes and sizes. And yes, just in generally, so how do the individual characters impact their fate and as well as their risks and remediation? Also for PFAS, we know it contains so many different compounds. It's a generic term. So in terms of PFAS, how do the different, change sizes or the functional groups affect their, fate in the environment and their also remediation? Maybe Denise, can you comment on this first?
Sure, I'd be happy to. Thanks for the question. I think that it's a big, complex question that is not easy to answer in a simple answer, and I think it always is, it depends. It depends both on the material itself, as you said, with the shape, size, potentially chemical composition, but also on the environment that we're looking at. If we're looking at water versus soil versus air, the different properties and characteristics of the nano and microplastics may behave differently in those different contexts. So we cannot always say that smaller particles or larger particles move more or less in environmental systems. It depends on the context which it is in. I would say the same also goes for hazards in terms of the adverse effects to different organisms. We may be able to anticipate different types of adverse effects depending on the size, shape, polymer chemistry, on the size of the organism, if it is consumed with food or not, ingested versus inhalation. So I think that more information on the context also informs how the different properties of the materials may move through or impact environmental or biological systems.
Thank you so much, Denise. Okay, Dennis, do you have anything to add on this point?
Well, I think it's similar for PFAS as well in terms of the receiving, if it's water, what the media is, water, soil, and also the PFAS are inherently surfactants. So it depends on the tail group and the head group, and both impart properties that will really impact their fate. And actually, Yan could do a better job than I on talking about the chemistry and destruction, but also that really does impact their destruction.
Yeah, thank you so much, Dennis. Sorry. Yang, can you also, you did a lot of work on PFAS remediation. I'm aware of that. So do you think, can you give us some your comments on the individual character impact the remediation technologies?
A pleasure. So long chain. So from my experience, long chain PFAS usually can be easily handled, relatively easier. So they can be subjected to heterogeneous catalysis process and also removed by sorbent. But shorter chain PFAS are more mobile, and it's harder to be removed using GAC and resin. And the second impact will be the functional group. So in general, sulfonate PFAS are more recalcitrant than other structure. And there's another emerging concern about the volatile PFAS now. So like FTOH, fluorotelmer, alcohol, they are more volatile. So when it comes to thermal related process or even foam fractionation process, gas sampling of volatile PFAS are necessary.
Thank you so much. Yeah, this is my question. Champion, maybe you have more question to ask.
Yeah, thanks, Shu. So I will start taking few questions from the audience, and I will start with Yang. So in extension of what you said about the treatment of PFAS, we have a question from Lubana. They are asking that could synthetic biology or CRISPR method be used to create bacteria capable of degrading PFAS? So
This is a question to Denise, not me. I'm not a biological guy.
Well, I'm not really necessarily a pure biologist myself either. I can maybe answer from the best of my knowledge about this question in regards to micro and nanoplastics, at least, that plastics, conventional plastics, are oftentimes very crystalline and then therefore hard to degrade by microbes because they can't access the carbon or break the carbon-carbon bonds. But different plastics can be developed to be biodegradable, and they are then more easily processed by these microorganisms. Yet that being said, this biodegradability isn't an overarching biodegradability in every context. It really depends on where the plastic ends up. So if it's developed to be biodegradable, for example, on an agricultural field, that has a certain pH, certain temperature, certain microbiology that is living in this field. And if that same plastic were to end in the ocean, it may not be the case for that same plastic because we have a different environmental context. I'm not an expert on PFAS, but I could imagine that some of the same context would also be true, that it would be challenging to maybe design a bacteria that would be able, or microorganism that would be able to degrade PFAS in a vast array of different contexts. And then we would also need to think about how these genetically engineered organisms are delivered into the environment to then degrade specifically this environmental contaminant, because we wouldn't necessarily want to be developing this type of genetically modified organism to then be spread globally just to degrade PFAS or to degrade microplastic. So maybe in very specific contexts, in treatment contexts, this may be possible, but probably not on a very large environmental scale, either for microplastics or for PFAS.
Thanks, Dennis. So I will go back to Yang again from for another question from our audience, that is there any risk of secondary pollution resulting from these degradation techniques that you spoke about, like DET or photochemical reduction processes?
The answer is yes. So we have to make sure, that's why I keep talking about treating PFAS should be in a regime of non-potable water treatment. So take the electrochemical oxidation as an example. If you apply a very high potential, then you will definitely form perchlorate. And perchlorate, but keep in mind that we are destroying a very recalcitrant chemical in exchange for a formation of secondary pollutant that can be relatively easier to handle subsequently. So this is like a seesaw structure. If you solve the PFAS problem, inevitably there will be new problem emerging, but luckily the problem usually could be, I would say, easier.
Yeah, thanks, Yang. Dennis, do you have something to add on this?
No, because PFAS is not exactly my area of expertise, so I leave that to Dennis and Yang.
Dennis, do you want to add something?
Well, no, there's always a concern, I suppose, with any treatment technology, and that you have, and certainly with PFAS, that you have complete mineralization, I guess, and that's a real. More and more, the scrutiny is going on to make sure that you are doing that, and we're not inadvertently creating PFAS that we don't detect, and that could be a future legacy problem.
Thanks, Dennis. So I will go back to Dennis again before I hand it over to Shu for another question from our audience. What are the current consensus views on microplastic transport pathways within living organism, and does it has any relationship with shapes and sizes?
I guess the difficult part of this question is consensus. So I think that it's challenging to get consensus from any group of experts, especially on a very new topic like microplastics or PFAS. I think that we're still in an exploratory phase about which are the most important characteristics. But again, as I had mentioned in one of my earlier answers, I think impacts to living organisms can be at different scales. This can be food dilution by microplastics, but it could also be crossing biological barriers and creating different internal harm. So I think then it depends on which types of hazards that you're looking at, where the size and shape would play differential type of impacts.
So I have a personal question. Like, we all know nanoplastics are most difficult to detect. So what are we now? I mean, do we have advanced methods to detect them more accurately than previous methods?
I think that this is always a ongoing challenge to be able to more accurately quantify nanoplastics. Here again, I don't think that we have the silver bullet in hand to say that this is the best way to be able to measure nanoplastics. And even in the case of microplastics, there are not yet standardized and harmonized techniques across the field. And these are even quote unquote easier to be able to measure than nanoplastics. So I think it would be great for the field to already have some standard analytics and harmonized analytics for microplastics. And then this may help us to be able to eventually inform that of nanoplastics, keeping in mind the analytical tools that are suitable to be able to measure microplastics may be different than those for nanoplastics because of their smaller size.
Thanks, Dennis. I will pass it to Shu now.
Thank you, Shampna. So yeah, we have great question from the audience, and one of them is from Manisha, and she has got two questions, and the two questions are also quite what I'm also curious with. So we know that nanoplastic microplastics can be a vector for other pollutants. Yeah, so that might also affect the transport and fate of the other pollutant. So the first question is, can presence of PFAS affect setting rate of micro and nanoplastics? This is addressed to Denise. And another question is addressed to Yang. Can microplastic impact degradation efficiency and energy requirements for degradation of PFAS? And if yes, what do you think in a way, in negative or positive way? Thank you. Maybe Denise, you can.
Yeah, sure. I'd be happy to take this one on. So in our settling studies, we had looked at the heteroaggregation between microplastics and algae and saw that the microplastics were acting as a ballast for the freshwater snow. So the freshwater snow is less dense, microplastics are more dense, and helps to increase the settling rate of the freshwater snow. When we're looking at microplastics alone, plastics overall are really a heterogeneous group of materials, and therefore microplastics are too. So sometimes PFAS would be present or not. We can also have different metals or different additives that are within plastics. But the main question is the density significantly different with and without PFAS? And I would envision that at the concentrations that PFAS is typically put into plastic, it wouldn't significantly change the density to change the settling rate of the particles.
Okay, thank you so much, Denise. Do you have anything to add, Dennis, on this one?
Yeah, no, that's great.
Okay, thank you. Yang, can you answer that question from our own?
Yes.
Okay.
So I think there are only a handful of evidence that PFAS interacting with microplastic. It's a emerging area, a lot unknown. I can only answer, by imagination. If you introduce a third phase, we talk about catalyst and water phase, right? But if you introduce a third phase where microplastic retains certain PFAS, and I think it will make the PFAS degradation more challenging.
Okay, thank you so much. Yeah. So, I really have one question regarding the regulations also, because we all know that it's getting serious on those, especially PFAS. I know there are quite a strict regulation going on here in the in the US or in other places around the world. So then both the US, for example, and EU have introduced strict limits for PFAS, and there is ongoing effort to create a global plastic treaty too. So, can you please talk more about this? Maybe Dennis, you can you have any comment on this one? Oh, sorry, I'm supposed to say Dennis.
Dennis
Okay.
from Australia. Okay, good. I was going to say
Yeah.
maybe he's the guy to ask.
Right.
It's a
I was so
confusing one
confused.
today as a moderator, I know.
Dennis, please, sorry.
You're doing great. Yeah. So PFAS regulations are changing at a really quick rate. And actually, in fact, today, Australia adopted new guidelines for drinking water, PFAS in drinking water. And undoubtedly, in the coming weeks, it'll create quite a bit of discussion. And I guess the question for me is it's related to drinking water, but drinking water is only one and maybe not the major source of PFAS to the human body. So there's a lot of focus and perhaps undue attention on drinking water. And what that does to many consumers in Australia, and I'm sure around the world, is that people are afraid to drink the water, which drinking water from the tap, which is mildly ridiculous because the risks of not drinking water vastly outweigh the benefits, I would suggest.
Okay, thank you. Yeah. So Yang, you would like to add something on that?
Yeah, so from US perspective, I think there are a lot of story behind it. But as of May 14 this year, we have PFOA and PFOS has those enforceable maximum contamination level. And I think another major development in this area is that now PFOS and PFOA as a chemical were defined as a hazardous substance in the United States. And any product containing PFAS need to report to EPA. That just for reporting this regulation.
Okay, thank you. Denise, right now your turn. Maybe some regulation going on here for nano and microplastics.
Yeah, so there has been a ban in the EU to have a ban on primary microplastics that are used in a lot of different consumer products and different applications. These applications can range from scrubbers and cosmetic beads to seed coatings and fertilizing agents that are applied directly to the field. So this has a few different phases of when these different products will no longer be allowed. It is great that we can reduce the amount of plastics that are in the environment when we look at having these bans. But on the other hand, the amount of microplastics that are coming from these primary microplastics is very small in comparison to the amount of nano and microplastics that are produced in situ in the environment from mismanaged waste. At the same time, reformulating a lot of products to be essentially polymer free will also be quite costly. So I think it's always a question of balancing what is causing environmental harm and the cost going into technological innovations to move away from polymer-free compared to using those resources in another way to help reduce mismanaged plastic waste, or moving more towards circular materials or finding alternative materials further up the life cycle. So there are also some suggestions to be having micro and nanoplastics consideration in the ongoing plastics treaty, but that's still under negotiation. So I don't have any further information on that point.
Okay, great to hear that. Thank you. Right now, I will hand this over to my colleague, Shumpner. Please, Shumpner.
Thanks, Shu. So we have another question that may sound a bit counterintuitive, and this is to you, Denis. The question is, the SAPE's evidence review report on micro and nanoplastic pollution published in January 2019 says the best available evidence suggests that microplastics and nanoplastics do not pose a widespread risk to humans or environment, except in small pockets. But that evidence is limited, and the situation could change if pollution continues at the current rate. Would you agree?
So it's hard to say if I agree or not in a direct statement if I have to give a yes, no. So as usual, I will give a contextualization because it's always hard for scientists to say directly yes or no, right? I think at the same time that this SAPEA report was released, there was also a similar report by the World Health Organization that gave slightly different wording that suggested that there's a lack of evidence to really be able to assess the risk at all. And so I think that this is rather if you want to prove harm or prove safety, and those are two different sides of the same coin. To be able to get some more foundational evidence about which is really correct, we need to, at least in the terms of hazards, be able to conduct studies that are really giving EC50s, LC50s, other different types of metrics that will start to let us know which types of materials in which conditions are affecting the most sensitive organisms. And I think that these sort of dose metrics aren't yet available.
Yeah, thanks, Dennis. Dennis, would you like to add something to this?
No, thanks.
Okay, thanks. So another question we have on PFAS, and I will go to Yang on this. Thanks for all the important discussion. Thanks, Drew. What advice would you give to policymakers trying to deal with PFAS and micro and nanoplastics who have introduced regulatory limits and bans, as you just mentioned, to not overburden communities who must pay their treatment or removal but were not directly responsible for the presence of these pollutants?
So there are two sides. So who is going to pay the bill? So in the United States, we have the Superfund mechanism. So who create that pollution pay the bill for remediation? That's already there on the table. As for putting myself in the shoes of policymakers, so I think the policymaker will make a critical role in screening alternative PFAS. So by pushing other alternative substitute PFAS, we want to make sure that there's there will be comprehensive toxicology study of these substitutes, and also we want to make sure that there will be degradation study to make sure that the degradation of PFAS in natural environment won't create all these legacy PFAS. That's my 2 cents.
Okay, thanks, Yang. Dennis, what is the situation in Australia? Do you have any information how the government is planning to tackle these things?
Well, so Drew has great points, and it's Superfund might be difficult. So I think inherently, I think that the polluter should pay. I think it's really important that what we don't do is point fingers at utilities, because utilities don't make the problem. The landfill operators or the wastewater treatment plants, they're receptors of society's wastes. So we have to be careful in that regard. And I would maybe suggest something slightly different than what Yan said in terms of I think that we need to, as a society, we need to carefully think of the chemicals we use in our day-to-day lives. And just because we can use a high-end product, let's say, for example, I mountain bike, I don't need to use high-end lubricant in my bicycle. There's many alternatives that are much less or much more environmentally benign. So I think that policymakers can help, but society needs to more broadly help and really push and have some consideration for that. But PFAS is a legacy issue, and there's certainly not an easy answer for that. But I think we have to be careful with pointing fingers at utilities, I guess is something that I would say.
Thanks, Dennis. Dennis, what is the situation in Europe that you are working for so long there?
Yeah, well, instead of discussing the situation specifically in Europe, I think I would take it in a slightly different mode. I mean, as Drew also mentioned in a follow-up of his question, that superfunds are often difficult to tackle or assess because it's a really diffuse pollutant source. But I would, at least for the case of nano and microplastics, at least already seeing how the current wastewater and drinking water technologies are at dealing with these different pollutants, and seeing if there can be specific upgrades or augmentations to current working conditions that would have better removal efficiency. So sometimes this is not necessarily going to be a change in infrastructure or even a change in costs. It's just changing the operational conditions in order to remove at least particulate contaminants. And this may be one way to move forward to see if we can just change what we already have to be able to better remove some of these materials from our water streams.
Thanks, Dennis. Over to you, Shu.
Okay, thank you. So we are approaching the end, but before we wrap up, just have one last question for me. Bear with me for a minute. So, and then we are now talking about the PFAS and then nano and microplastics, and then we know that they are causing problems. So when we think that maybe we find alternatives, and that's what I think we are doing right now. So yeah, please, if it would be great, you can elaborate a little bit more on that. And Denise, I know you talked about something about non-polymer, products. And also, how do we make sure that those alternatives are not introducing toxicity? For example, the alternatives to PFAS. Okay. If Dennis, could you please start on this one?
Yeah, well, I don't have a specific alternative in mind, given that there are so many different uses of plastic and even primary microplastic that are used in different products. But in the EU, there is a new push towards safe and sustainable by design of materials. So this assesses the material as it is being produced to not only look at the toxicity, but also life cycle assessments and other key things that can negatively impact the environment, not only during the use and end of life phase, but also looking at the beginning of life throughout the entire life cycle. So I think taking more of a safe by sustainable design approach and thinking holistically about the product from the beginning would be a helpful step in this direction.
Okay, thank you. Dennis, can you tell us something about this too?
Yeah, so I agree with Denise. We have to start thinking holistically. And with regards to PFAS, there's a range of initiatives, but I'm not sure that it would be considered holistic. For firefighting foams, they've gone PFAS free, so there's no fluorine in there. And the thought would be that it would be more environmentally benign, but still effectively put out fires. And then there are bans in Europe and in Australia with the amount of PFAS coming into the country or encapsulated in consumer products. So those are some initiatives. But I think sort of PFAS is quite a broad group of chemicals, and they were put into products for a reason. So we have to make sure is that the alternatives that we put in there, those functions, that we don't inadvertently create another long-term legacy problem that we're not properly considering at this time.
Yes. Okay. Yang, would you like to comment on that too?
Yeah, so Professor Ian Cusson from Stockholm University published a cool paper early this year. So in that paper, he said that there roughly 10% of PFAS have already have a safe substitutes. That's a promising sign. And then US, as for in the United States, DOD have a lot of effort in developing fluorine-free firefighting foam. And it's a fun fact to share that I heard from a video yesterday. There's a soybean derived firefighting foam that has an excellent performance compared to PFAS containing HFF. So definitely, everybody's. This is a hot topic, and we definitely are seeing some hope.
Yeah, thank you so much, everyone. Right now, I will hand this over to Fabio.
Well, I will always like to thank everybody again. Thank you, Denise, Dennis, Yang, for your invaluable contribution, and thank you, Somaparna and Shu, for organizing this. And I would like just to say that this is actually a very, very complex topic, and there will be a lot to discuss. So please just follow the field as you can, follow the speakers who are, and their work, who are doing great work in these areas. I will just put briefly the QR code again for the collection, but in the meantime, I would like to also say goodbye and hopefully see you next time. Thanks everybody for coming, and hopefully we'll see you again at the next installment of this series. Thanks everybody again.
Thank you.
Thank you.
Thank you so
Thank
much,
you.
everyone.
Take care.
Everyone.
Bye.
Thank you.
Bye.
Bye. Bye.